So again, today we're talking about cell membranes and cell transports. You're going to be able to explain cellular processes, including homeostasis and the transport of molecules. All right, so what do you know about the cell membrane? I've talked a little bit about it before during all of our lessons. So type in anything that you can remember about the cell membrane. I hid your name, so don't worry if you put the wrong answer. It is made up of lipids. Good job. It is the outer layer of the cell. Good job. It lets things in and out. Yes, that's good. Good, good, good. It regulates what enters and leaves the cell. Yes, that's correct. It lets things in and out and can be fooled by viruses and bacteria. Yes. It's semi-permeable. Lets things in and out. Yes, yes, yes. It lets things in and out. Controls what goes in and out. It's the cell's boundary. Monitors what it leaves and enters the cell. These are all great answers. Good job. So you do remember quite a few things about the cell membrane. Good jobs. Yes. Good job. Good job. Good job. All right. I love these answers. Let's carry on. So the cell membrane is kind of going to act like the door of a house. It's going to control what goes in and out of the cell, and it's going to keep everything protected on the inside. It's going to provide a protection, kind of like the skin on your body provides protection for all of your insides. That's what the cell membranes do. And then it's just going to hold everything in place. It's going to make sure all the organelles stay where they need to stay or move where they need to move. So we say that the cell membrane is a fluid mosaic model. We say fluid because it's not rigid. It's able to move how it needs to move, to go where it needs to go, or to accept what it needs to accept. It's fluid. And it's made of many components, so we say it's mosaic. It has proteins, cholesterol, and carbohydrates. So the proteins, cholesterol, and carbohydrates are all embedded somewhere in the cell membrane. That's why we say it's mosaic. There are two types of proteins. Right here we have the integral proteins. These are the large purple things. They go from the heads of the membrane to the tails of the membrane. We have peripheral proteins that are just on the heads. They're just located on the top. They don't go through the whole membrane. The cholesterols are going to be fatty acid chains that are embedded inside. You can't see any in this picture, but the cholesterols are there. They help with stability. They help with the strength of the cell membrane. They keep it sturdy. And then the carbohydrates, they're chains that extend from the proteins. And these carbohydrates are going to help identify the other types of cells and the other types of molecules to see what it needs to bind to and what it can accept. So this is a 3D version of the cell membrane. This is what it looks like. It has its heads. It has its tails in the middle. Notice that every head and tail is partnered up equally. And this surrounds the entire cell. So remember I talked about a basketball being a cell and a blanket covering up the basketball? This cell membrane would be the blanket that's going to cover the basketball. All right, so let's see if you remember. What describes the cell membrane? Which term describes the cell membrane? Again, your names are hidden. No one will be able to see your answers. Good job, good job. All right, so hopefully you remembered that it was fluid and mosaic. Does it tell you the answer after you click it? No? Okay. Well, it's fluid and mosaic. Remember, it's able to move around how it needs to, and it's mosaic because it's composed of many parts. It does allow things to go through it. It's not rigid. It's not sturdy. It is able to move. A cell wall would be rigid. Good job. And no one picked D. Good job. So we call the cell membrane semi-permeable. That means it's going to let some things in and it's going to keep some things from coming in, which is great because we don't want everything going into our cell. We want to make sure we maintain homeostasis. That's why we say it's semi-permeable. If it has already enough sugars, enough calcium, enough potassium, it's not going to let anything else go inside of it. If it needs more sodium, if it needs more salt, if it needs more sugar, it's going to let those things in as it needs it. That's why we call it semi-permeable. Some molecules are permeable, meaning they're allowed to go in through the cell at any entry point. And then some molecules are impermeable, meaning they're not allowed to go in. The cell membrane will not let it enter at all. And those are going to be the larger molecules. So we say that the cell membrane is a phospholipid bilayer. That means it's a double layer of lipids. And if you remember, lipids are fat molecules. So the phospholipids have phosphate heads. These are the top parts, the outer parts of the membrane. And the heads are hydrophilic because they love water. Think of philic, likes water. Philic likes water. And these are the parts of the cell that are able to stay within the moisture area, again, the water area of the body, of the inside of the cell. The tails are the lipid parts. And the tails hate water, so we call them hydrophobic. How many of you have a phobia? Does phobia mean you love something, or does phobia mean you're scared of something and you hate it? Hopefully you know that phobia means you don't like it. You don't want anything to do with it. So the lipids are hydrophobic. They're afraid of water. They don't want anything to do with water. If water goes into the membrane at all, it's going to repel it quickly because these lipids hate water. Tomorrow I'll show you what it looks like when we mix oil and water together, and you're going to see that they hate each other. They're going to push apart as much as they can. All right, so go ahead and label the hydrophobic and hydrophilic region. Also label which one's the head and which one's the tail. We just went over this, so hopefully it's fresh in your head if you're paying attention. Good job. All of you are remembering the pink part is the head, and that is hydrophilic. Good job, good job. Y'all are all putting great answers. Some of you aren't doing anything at all. Guess you're just sitting there. And then y'all are remembering that the tails are the hydrophobic part. That's the yellow part. Good job. All right, this is great. So I'm glad that you understand the difference between the two. I'm going to keep going because most of you already wrote down everything and it looks great. All right, so in your notes today on your Schoology page, I left you a link to a membrane simulation. This is what it looks like. And if you click on the words, it's going to tell you the definitions and a little bit about them. This is a great example of the cell membrane, a great tool to study. So I highly encourage you to look at this whenever you have time today, after you're done with the notes, after you're done with the lesson. So now we're going to talk about types of cell transport. And cell transport is the movement of particles across the membrane. And again, we want to move molecules to maintain the homeostasis. We want to keep things balanced and checked. So we're going to be moving sugar molecules. We're going to be moving salt, sodium, potassium, calcium. We're going to be moving those through the membrane to make the cell happy. We are going to have passive transport, which requires no energy at all. It's very easy to move those materials. And that's going to be like riding your bike down the hill. You know how easy that is. You don't even need to pedal. You just coast your way through. That's going to be for molecules that go from a high concentration to a low concentration. And what I mean by that is, think about when you were back in school before COVID happened. Way before COVID happened. You're in the hallway. Everyone's in the hallway with you. You're trying to get to your next period. You know how crowded that is? And that's what we call a high concentration. Then when you slowly start going into the classroom, it becomes low concentration until it fills up again. But if you have like 1 or 2 people in the classroom, when you walk in, that's a low concentration. So high is the crowded hallway. Few people in the classroom is the low concentration. Active transport's opposite. Active transport's really hard to move molecules. It's so hard that it's going to need the help of energy molecules like ATP, which is energy. And it's going to be moving molecules against the gradient. So not high to low, but low to high. It's kind of like forcing your way into a crowd. If you've ever been to a concert, it's like forcing your way to the front row. You got to fight through all of those people. That's what active transport is. It's kind of like riding your bike up the hill. You need to pedal harder. You need to use all your strength to get up the hill. That's active transport. All right, so what's the difference between passive and active? Again, your names are hidden, so you don't need to worry if you put the wrong answer. What's the difference between passive and active? One uses a lot of energy, one uses no energy. Good job. Passive requires no energy and active requires energy. Good job. Passive is easier to do and requires less energy. Good job. Active is difficult and passive is easy. Good job. Passive is from high to low concentration while active is the opposite. Yes. Passive uses less energy. Yes. Passive uses no energy while active you need to use energy. Yes. Passive transport is less energy being used while active transport is more energy. These are great answers. Good job. Passive goes with the concentration, active goes against. Yes. Passive uses very little or no energy, active uses a lot. Good job. Great answers, guys. All right, so again, we're talking about concentration gradients. With concentration gradient goes from high to low because it's always easier to go from high to low. It's always easier and more comfortable going from a high crowded area to a low spaced out area. Active transport is going against the concentration gradient. Again, we're going from an area of low, easily spaced out, to an area of high, pushed together molecules. All right, so go ahead and match the type of transport to its description. You could try again if you get it wrong. Good job, good job. So again, passive goes from high to low, active goes from low to high. Passive goes from high to low, active goes from low to high. All right, so there are 3 types of passive transport, and we're going to get into more detail tomorrow. But diffusion is when the molecules are so small that they can actually go through the lipids. They're still going to go from high to low, but they're going to travel through the lipids. Osmosis is the water molecules. Water molecules are always going to travel from high to low. And then we have facilitated diffusion. Since these molecules are bigger, they're going to need the help of a protein pump, but they're always going to go from high to low. All 3 of these will move from high to low. Then we have active transport. Again, active transport requires energy. It's going to need the help of ATP, active transport, ATP. Because it's going from low to high, it's going against the gradient, so it's definitely going to need to use the protein pumps. The ATP, and it's going to kind of act like a key. It's going to open up the pump so that way the molecules can move through, going against the gradient. Now, sometimes we have very large molecules that are so large they can't go through the pumps. They can't go through the heads of the lipids. They can't go through the pumps of the proteins. So we're going to need to engulf them. The cell's going to need to surround it and bring it in. So if you remember when we talked about the endosymbiotic theory, I said a large bacteria ate smaller bacteria, and it engulfed it. It brought it in. That's active transport. It needs energy to do that. It needs energy to use the membrane to surround something and to pull it inside of it. That's what we call endocytosis because it brings it in, endocytosis. Then we have exocytosis, which does the opposite. It makes the membrane around it inside of the cell, and then it pushes it out of the cell. I have a lot of videos to show you these tomorrow when we go over transport one more time. But for now, go ahead and fill in the blanks to this assignment. And then after you're done, you're free to go. If you do not have any questions after this, you are free to go. If you have questions, stay on and I'll be happy to answer them.